Evolutionary patterns and processes. Learning objectives. Explain scientists' hypotheses about the origin of life. Describe the theory that explains the origin of eukaryote cells, and explain what fossils can reveal about ancient life. Researchers have made major progress in understanding the origin of life. Nobel Prize-winning origin of life researcher Jack Szostak has written that the exact circumstances of the origin of life may be forever lost to science. That may be true, but experiments have shown that the complex molecules like RNA can be formed in the absence of life, replicated, and carry information. The results of experiments like those with RNA give many scientists confidence that Szostak has also said research can at least help us understand what is possible. In 1953, chemists Stanley Miller and Harold Urey tried to test the hypotheses that organic compounds could have been produced on early Earth. They warmed water in a sterile flask and added methane, ammonia, and hydrogen to simulate what Earth's early atmosphere might have been like. They passed sparks of electricity through the gases to simulate lightning. The liquid circulated through the experiment apparatus for a week. The results were spectacular. Miller and Urey analyzed the results and revealed that 21 amino acids had been produced in their apparatus. A more recent analysis of one of Miller's experiments not only confirmed the result, but also found the experiment had produced 2 additional amino acids for a total of 231 amino acids. Miller and Urey's experiment suggests the organic compounds necessary for life could have arisen from similar compounds on primitive Earth. While Miller and Urey's hypothesis about the composition of Earth's early atmosphere were incorrect, more recent experiments using current ideas about Earth's early atmosphere have validated their conclusions. Organic compounds could have been produced on early Earth. The discovery of organic compounds on meteorites and comets has further confirmed these lab experiments. Several lines of evidence suggest that RNA rather than DNA was the first information-carrying molecule. The RNA world hypothesis proposes that RNA existed before DNA. From this simple RNA-based system, several steps could have led to today's DNA-directed protein synthesis. This hypothesis is still being tested, but evidence makes it credible. Recent experiments show that at least 2 RNA nucleotides can form in the absence of life. Other experiments show how simple clay minerals can bind to nucleotides, which can polymerize to form RNA polynucleotides. In addition, certain RNA sequences are capable of catalyzing chemical reactions that include RNA self-replication. Furthermore, several molecules important to cell metabolism, such as ATP, NADPH, and coenzyme A, are built around RNA-like nucleotides. All cells make proteins, but first copying genetic information into RNA, DNA nucleotides themselves are first synthesized as RNA nucleotides. Finally, the ribosomes that build proteins in all cells are RNA machines, in which RNA, not DNA, performs the key function of linking amino acids together. The biosphere as a force of planetary evolution. Microscopic fossils of prokaryote cells have been found in rocks more than 3.5 billion years old. When these first life forms evolved, and for more than a billion years afterwards, Earth's atmosphere contained very little oxygen. Then, during the early Proterozoic era, roughly 2.2 billion years ago, photosynthetic Earth evolved. The evolutionary event in the biosphere had profound consequences for the atmosphere, geosphere, and hydrosphere. Some biologists suggest that the evolution of photosynthesis was the beginning of the full interactive relationship between living organisms and all the aspects of their physical environment. Early photosynthetic bacteria began to chum out oxygen in a steady increasing quantities. Oxygen is a highly reactive gas which was not present in Earth's early atmosphere. As its production and release was dramatic enough to have effects, oxygen combined with iron in the oceans, producing iron oxide, which is rust, that sank to the ocean floor and is now the source of most iron ore mined today. You can see bands of iron oxide formed billions of years ago. Next, oxygen accumulated in the atmosphere, forming the ozone layer. Over several hundred millions of years, oxygen concentrated in the atmosphere, increasing to the points where it drove some early anaerobic life forms to extinction. Other organisms, however, evolved new metabolic pathways and used oxygen for respiration. These organisms also evolved ways to protect themselves from oxygen's powerful reactive properties. Eukaryote cells, as you've learned, contain several kinds of complex cytoplasmic organelles, including lysosomes, endoplasmic reticulum, cilia, and flagella. Two other organelles, chloroplasts and mitochondria, are even more complicated. The evolution of these complex eukaryote cells from much simpler prokaryote cells set the stage for the rest of the history of life. How did the cellular complexity evolve? The earliest eukaryotes were the earliest to evolve. The fossil record provides few clues to the evolution of cells. The oldest eukaryote cells fossils have been found in rocks 2.1 billion years old. Unfortunately, these microscopic fossils don't usually show details of internal cell structure, so they provide few clues about early eukaryote evolution. The endosymbiotic theory, developed in the early 1960s by Lynn Margulis of the University of Massachusetts, proposes that organelles and eukaryote cells were formed when different types of cells joined in a kind of merger. Margulis called her idea endosymbiosis, which literally means living together within. A great deal of evidence from molecular biology now supports the theory that many of the complex features of eukaryote cells evolve through endosymbiosis. The fact that some cells today contain bacteria and algae living as endosymbiosis further supports this theory. Mitochondria, for example, are similar in size to bacteria, have their own DNA genomes, synthesize some of their own proteins, and are formed from pre-existing mitochondria inside dividing cells. From these and other reasons, most biologists now agree with Margulis's ideas that mitochondria are the descendants of free-living bacteria that took up residence in early eukaryotes. Similar evidence supports an endosymbiotic origin for chloroplasts. Chloroplast membranes resemble those of protosynthetic prokaryotes. Chloroplast DNA have many similarities of prokaryotes as well. This suggests that chloroplast evolves from pre-existing and pre-living photosynthetic cells that paired up with early eukaryotes. What can fossils reveal? Fossils reveal information about structures in ancient organisms, the sequential nature of groups in the fossil record, evolution from common ancestors, and the ecology of ancient ancestors. Comparisons of body structures can be used to test hypotheses about the appearance, evolution, and history of groups in the fossil record. Studies of evolutionary change in the body structures can also be used to test hypotheses about the evolution of living species from the extinct and common ancestors and the evolution of diversity.